And if we look at the sort of things we'll be doing, that's what you'll see how that relates. And yes, I was going to call this the colour purple as well, but I thought, oh, maybe not.
Can I get you to just use this to change your slides?
Okay, wonderful. Okay, getting an academic to use modern technology. That could be interesting. Okay, that was the first one, wasn't it?
Yeah.
Okay, this is obviously a staged photo because my students do not normally look so enwrapped when I talk to them. But I'll get it right eventually. How do I go back? I've worked it out. Is the number of years of healthy living with some disability and then with severe or profound activity limitation in that age group that are now reaching 65. So that's an interesting group, because if we can try and either prevent the onset, if we can extend that green period, then we're certainly going to improve quality of life. And can we do that with food in particular? So can we use the components of food to improve life expectancy? Now, we all know the sorts of things that the risk factors, and those risk factors are fairly, in a large proportion of the population, and one of those, I think, is standard around the westernised world, the westernised diet: not enough vegetables, not enough fruit, and also not enough exercise. But what that will lead to is very clearly down the bottom there, the cost of providing healthcare dramatically increases in that population from about 65 onwards, dramatically increases. And so there's clearly a lot of possibility to improve that. Now, that data comes from the Australian Institute of Health and Welfare, so AIHW, which is a standard statistics gathering arm of the federal government, of our federal government. So, a hypothesis, which I think may even go over to the second and third speaker this morning, that food bowls, and clearly this area is a food bowl as well, so Manitoba, can become a major producer of functional foods in the treatment of obesity. And just some numbers down the bottom there, the wheat production in Australia is about 30 million tonnes a year. The wheat production in Canada is about 30 million tonnes a year. And so we have very similar in that way. But the key thing with any of these is to provide the evidence, and not simply say, hey, that sounds like a really nice idea that a purple food is good for us. I was at one of our local schools 10 days or so ago with a grade 7 class and asking them, gave them some purple carrots and asked them, and they ate them all, and asked them, how would you prove they're important? And those kids actually came up with the idea of what to test and also to use a control. Now, if only we could get most of the population to be as smart as most grade 7s. But the key thing with that is providing the evidence. And the other gentleman in the photo, the rat is obviously the important part of the photo because that's where we do the experiments. And the other gentleman in the photo is also here, Sunil Panchal is also here. He's decided not to listen to me once again, but to go to the microbiome session next door somewhere, because he thinks that's going to be more interesting. But he's done a lot of the work. If you see him around, you can ask him questions. He will know much more about the experiments than I do. But providing that evidence is the key thing. There is that, if you can read it down the bottom, there was a Landline program. Landline is the Australian Broadcasting Commission's Rural Affairs program every Sunday. I've watched it once in my life because we were on it, but apparently everyone else in the nation seems to watch it. But this was looking at the extra purple plums, and so if you want to look at that, obviously, if you Google the colour purple and ABC, you'll get it. So what do those foods have in common? Purple carrots, the Queen Garnet plum, purple maize, seaweeds, linseed, flax. I've got to get the right terminology. And of course, last but not least, coffee, real coffee, is also interesting. So what do they have in common? Of course, the first thing people say is antioxidants. That's probably the last time I'm going to use that word in this talk, I hope, because I have no idea what that word actually means. It's a great marketing word. Everyone seems to have an understanding, and they're all different. So, and I don't really know that most of the things we deal with are oxidative stress diseases. I'll talk much more about anti-inflammatory. So what we do is use rats, and we treat them for 16 weeks. So over 16 weeks, we measure blood pressure, we measure oral glucose tolerance, abdominal circumference, insulin tolerance, DEXA, echo, and then a whole series of experiments on isolated tissues or on plasma. And we'll go in always to over 16 weeks, those rats will get fed a high carbohydrate, high fat diet. So they're getting 20% beef tallow. They're also getting about 40% condensed milk, and they're getting 70.5% fructose in the diet, and in the water there's 25% fructose. So why have we used a high carbohydrate, high fat diet? Because that's what the westernized diet is. It's not simply a high fat diet, it's not simply a high carbohydrate, it's a combination of both. We don't use the genetically obese animals, so the Zuckers, for example, for one very simple reason. Humans aren't genetically obese. They're diet-induced obesity in most cases. And so if we want to mimic what's happening in humans, and therefore possibly get an intervention that can translate into humans, we need a similar model. Now, we also go in and intervene. That was with seaweeds, which I'll talk about later, but we intervene from 8 weeks onwards. So they've had 8 weeks of that diet, they're already overweight, if not obese, their blood pressure is up, all of these things, and then we go in and intervene. Why? It would be a lot easier to do a prevention protocol. But unfortunately, patients don't go to their doctor 10 years before they're going to become obese and say, give me a treatment to stop the obesity. They go when they're already obese, and when the family has said, we want you around for a bit longer, go and do something about it. So if we're going to mimic the human condition, we need to do so in a model. And if we look over the 16 weeks, the blood pressure of these rats goes up from systolic blood pressure of about 110 to 150 to 160. And it's a consistent rise. So you can see the 8 weeks there, it's already over 140. The CS is a cornstarch diet, so it's the control one. If we look at the body fat mass, so these are DEXA results, at 8 weeks and at 16 weeks. So 8 weeks and 16 weeks. So the lean mass in grams there doesn't really change much. Goes up a bit in the 16-week fat ones, but doesn't really change much. But the fat mass goes up from the standard about 60 grams in the control animals up to about 160 grams. So that's about 20% of body mass increase as fat. So we need to get onto something purple. Now we've had all that introduction. And so in particular, the purple carrots. Now the purple carrots apparently are available here. I'm sure you can tell me that better. So these are the carrots that are purple the whole way through, not just purple on the outside, but purple the whole way through. And the question we'll always get asked is, are they genetically modified? And the answer is very simple. No, these are actually the original ones. The orange carrots are the really new one. The orange carrots have been around for something like 500 years. The purple ones were recorded in literature at least 5,000 years ago in the Middle East. And then there's red ones with the same colouring agent as tomatoes, yellow carrots with lutein, white carrots with nothing, so no colour at all, and not surprisingly they come from Western Europe. And then the orange carrots with the beta-carotene. And if we look at the results, so if we put purple carrot juice, so these carrots were juiced, and then that juice concentrated, and so that juice contains mainly the anthocyanin, so that anthocyanin in particular, and very little else. So So very little carotenoids in that because they're lipid soluble. So if we crush the carrots in water, we don't get that. It stays with the pulp. And if we give that at 8 weeks and we compared it with beta-carotene, both of those lower blood pressure. Didn't get quite back to control, but it was vastly different from the untreated. So this is a high carbohydrate, high fat diet with those interventions. So we're not changing the diet in any way. We're simply adding this to it. And it's brought that blood pressure significantly lower. If we look at the abdominal fat pads, the fat diet doubled approximately the fat pads. And again, the last 8 weeks with purple carrot or with beta-carotene has brought that back to normal. So what I will emphasize time and time again, probably you get sick of me saying it, this is not a billion-dollar multinational company white powder. This is a carrot. This is the color from a carrot. And that's all it is. So is this a drug or is it a food? There's no real answer to that, obviously. If we look at the plasma lipids, so cholesterol, triglycerides, and free fatty acids, again, in these rats, we see an increase in the fat diet, and we see a decrease with cholesterol, almost back to normal with triglycerides, but a difference there between the beta-carotene and that. So one piece of evidence that the beta-carotene is not doing the same things as the anthocyanins. Beta-carotene is, of course, the precursor for vitamin A, and that's a totally different aspect. If we look at the cardiac structure and then function, that lighter red is the staining, picoserous red staining for collagen. So these fat rats have enormous amounts of collagen around the cells. Whereas when we treat, look at that, there's virtually no collagen there. Same with the beta-carotene. There's virtually no collagen left. So collagen can be removed from the heart remarkably rapidly. Even though we're all taught in undergraduate that collagen stays there forever, it doesn't. But we look at a functional measure, and this is the Langendorf heart. Now, the Langendorf heart is not new, it's not novel, it's not innovative. It was published in 1895. And a lovely paper, it's about a 30-page paper in German, but a lovely paper explaining what they did, what went wrong, how they learned from it. And so not this, these very short papers that we now write, but really interesting to read. And if we look at the stiffness of the heart, so balloon put in there and measuring the left ventricular stiffness, the normal heart is about 20, 21, and that goes up to 28 to 30. But look what happens there. The purple carrot extract fed rats, still on that awful diet, have a stiffness of the heart back to normal. So a functional measure of the ability to relax, of the ability to contract, to expel blood, back to normal. Beta-carotene didn't do that. What about the liver? Now, all those different colors simply means that we stained them on different days. It's not that we didn't actually have purple livers because we gave them purple carrots. It is the stain. But in the high carbohydrate, high fat, lots of that lipid droplets in the liver, lots of inflammatory cells around the blood vessels. Purple carrots, where are they? The lipid droplets are not there. It looks as if the cells have been, from when we looked at the molecular biology, the cells have undergone apoptosis and have been removed. It's not a regeneration of a normal cell from a damaged cell. It's new cells. And the inflammatory cells are also gone. This did not happen with beta-carotene. There's still the fat globules in there. That's a blood vessel. But the fat globules are still there. Inflammation's gone, but the fat globules are still there. So the anthocyanin, the color, is doing something that beta-carotene does not do. And that's why I would use the term antioxidant. We have anti-inflammatory, sorry, not antioxidant, anti-inflammatory. Inflammatory cells, no inflammatory cells. That, to me, is the mechanism. And we published that in the British Journal of Nutrition about almost 5 years ago now. At that stage, in Australia, the purple carrots were virtually unknown. Now they're in quite a number of the supermarkets. It's not, we certainly didn't predict that. There was something like 250 websites then that mentioned purple carrots. Within a year, there are 8,000. And so that power of, social media. Yes, we're on the media every so often, but that power of social media is amazing in this field. But is that the only one? I want to introduce the Queen Garnet plum. The Queen Garnet plum was an accidental cross by some of the scientists at our local Department of Primary Industries, and it has 5 to 10 times the anthocyanin content of a normal plum, and it's purple the whole way through. So there's no sort of orange core, it's purple the whole way through. So we've taken some of the juice of that and compared what happened. So again, 8 weeks onwards, the fat rats, the body weight keeps going up, and you can see the marked difference there when they have the same calorie intake, but with the purple plum juice. The blood pressure, back to normal. So the green one versus the red one. This is not a billion-dollar product. So if we look now at the fat mass on these, so if we look at, it's really hard to read the slides from this angle actually, but if we look at the fat mass on there, what we see, I can see better here. No, that's the stiffness up there. Okay, that's the stiffness on that one, so the stiffness of the heart goes up dramatically, as we saw before, back to normal. The total fat mass and the abdominal fat mass are back to normal. For the abdominal fat mass, there is a bit extra fat throughout the body, so there may be a redistribution of abdominal fat. But after only 8 weeks of treatment, it's effectively back to normal. If we look at the plasma lipids again, they're up in the fat rats, back to normal with the treated rats. And again, a measure of liver function, so ALT concentrations, plasma ALT activity goes up there, so there is damage to the liver, and it's back to normal. Food or medicine? And again, the histology, and we've got a blood vessel there, vast numbers of inflammatory cells in the liver. We don't see any. It's anti-inflammatory. Oh, and the other reason for putting another university's name down the bottom there was to remind me, and it almost didn't, that we have started now a small clinical trial with some colleagues at Victoria University, which is in Victoria, so it's in Melbourne, and looking at overweight, mildly hypertensive patients, and we'll be treating them for 12 weeks. The first patient should have started this week, taking their purple carrot juice, the purple plum juice, and let's see what happens with that. Is that the only purple one? No, there is purple maize. Now, we don't grow purple maize, purple corn, I don't know what the right word is here, in Australia, but it is one of the important ones from the Inca civilization. And when we've used that at the same concentration of anthocyanins, we get, not surprisingly, basically the same responses. So blood pressure back to normal, certainly a decrease in abdominal fat mass, certainly not back to normal yet, but this is only 8 weeks. And so maybe it will come down further, we don't know. But certainly a decrease there in both those key parameters. Chokeberry, we've done some chokeberry juice as well. And again, when we've used the same concentration of anthocyanins, so the same dose of anthocyanins to the rats, blood pressure back to normal, the abdominal fat or total body fat mass, abdominal fat mass similar, almost back to normal. So is that the only colour that is working? Well, we've looked at beetroot. Now, beetroot have some wonderful compounds in them, these betanes, lovely complex compounds that organic chemists get really excited about. And then they have also sodium nitrate, which is really simple, really straightforward. And does it work? Well, the end of the story is that the sodium nitrate works and the colour does nothing. So let's look at some of the data for that. So firstly, if we look at the body weight, and you can see all of those, so the one with the beetroot, the one with the sodium nitrate, you can see, hey, there's no difference. So it's not doing a thing to obesity. It is bringing blood pressure down, but both to the same extent. So the beetroot and the nitrate, both to the same extent. The fat and the lean mass are very clearly unchanged. So if we look at, so the fat mass goes up in the fat rats, but it's not changed by either intervention, and lean mass not changed by any of it. It does improve glucose tolerance. So if we're looking at the oral glucose tolerance test, an area under the curve, and with the plasma insulin there, but area under the curve, it's brought that back to normal, it's brought the increase. So these are a model of insulin resistance. It's a diet-induced insulin resistance. The insulin goes up, and it's brought those back to normal. But you can see that the beetroot and the nitrate are doing exactly the same things. And the plasma lipids, again, the cholesterol, the triglycerides, the free fatty acids are being brought down by both interventions to exactly the same amount. Now, we measured the amount of nitrate in the beetroot juice, and then we've dosed with exactly that amount. And if we look at the liver enzymes there, so again, ALT, AST, and ALP is the major ones, and you can see the increase there in the same scale, but the increase and brought back, maybe not always to normal, but certainly markedly decreased by both interventions to the same extent. And if we look at liver structure, again, lots of fat in there, no fat in both. So what we're seeing is exactly the same response to the nitrate as to the beetroot, suggesting that it really is simply the sodium nitrate. So beetroot are a wonderful thing when they're fresh, are wonderful, and certainly will add to cooking and doing all sorts of things, but it's the nitrate in there, not the colour. And the cardiovascular function we've measured as well. So if we look at these are from echo, so left ventricular, can't read that, LVIDD, internal diameter at diastole, and then the diastolic stiffness in that one, we can see they're back to normal, so we've got cardiovascular changes coming back, we've got the glucose metabolism coming back, but we're not seeing any change in fat mass. The left ventricular structure, again, all that collagen in the fat rats, back to normal. There are inflammatory cells in there in particular, there's none in there. The black dots are the nuclei of the myocytes. And of course, I couldn't come to Manitoba without talking about flax or linseed. And when we've looked at these ones are with chia oil, because chia is obviously very trendy. It's also 10 times the price, so obviously it must be 10 times as good, mustn't it? And so if we compared the effects on the rats with chia oil, we also did linseed oil, and it's the same response, and compared that with the same dose of DHA, and what we're seeing in virtually all parameters is exactly the same response. So at least in these fat rats, we're not seeing any difference between the different omega-3. So ALA, EPA, or DHA are giving basically identical responses. Of course, if you present that to a fish oil lobby, they will very impatiently want to question it very intently. But we're seeing the same effects. So we would argue that we do not need to get conversion of ALA to DHA to be ineffective, that ALA by itself is effective in metabolic syndrome. It certainly improves the liver structure, as you can see there. The fat, the chia oil or linseed oil both give the same effect. We get rid of the fat, we get rid of the inflammation. DHA does the same thing, not surprisingly. So are they the only things? Well, there's a whole range of possibilities. We've looked at a sea lettuce. This was work done with James Cook University in Townsville because they have a superb marine science research facility, and also they're very close to the Great Barrier Reef. And with these, if we look at that one in particular, the sea lettuce, what we see with blood pressure is coming back to normal, and we see a decrease in the fat mass. So this was work published in January, I think it was, January or February this year. But of course, we can't live by living on lettuce only, and so we need some spices in our diet, and one in particular is black cardamom, in particular from Sikkim, in northeast India. One of my students came from that area, and his father kept telling us about how wonderful black cardamom is, and so we decided, well, why not? We'll try it. And it actually does work, probably at a dose that is not relevant to the human diet, but there's ways around that. So if we look at black cardamom in the diet, the systolic blood pressure is back to normal, and if we look at the fats there, yes, they're back to normal as well. And last but not least, there must be something good about coffee. Not simply the CNS effects, but let's look at the metabolic effects. And sorry people, if you are drinking coffee because it's going to lose weight, no, sorry, it won't work. But it does improve liver function very dramatically, brings liver function back to normal. So about 6 months or so ago, our ABC radio phoned me up for an interview, and they just found this wonderful new paper from Harvard that was saying that coffee improves liver function. And I thought, well, that's really nice. That's good of them to know that. And so I mentioned that we actually published that in a fairly reputable journal, Journal of Nutrition, 2 years ago, or 2 years before that interview. We didn't get cited, of course. So I would suggest that food bowls can diversify, so simply not looking at commodities, but looking at ways to use the non-nutritive portions as health foods. Now, how are we going to do it? Are we going to have it as the original food? And of course, I think one of the most interesting foods at all is red wine. And yes, I'm totally biased, Australian red wine. But do we do it that way as the original food? Do we tell people to eat 3 purple carrots a day? I think the compliance with that is going to be, well, not with the red wine, but the compliance with the purple carrots is probably going to be 0. Is it better then to, and this is one of the companies we've worked with, is it better to have a concentrate so that when they take 30 mils and put it into a glass for their morning fruit juice, they've got their daily dose? Possible. So it's still certainly is it then a functional food? Is it a nutraceutical? I don't know. Or do you take this, as in this company's just put this on the market, where they've combined some of our work with elagitannins from the oak barrels that certainly go into the red wine, and also the purple carrot into a capsule. And again, take 1 capsule every day, 1 or 2 every day, to relieve joint pain or to reduce blood pressure. Clinical trials? Of course not. Anecdotes from patients? By the 100. Which one's more valuable? To the company, the anecdotes. To the scientists, the clinical trials. So there's always going to be a difference of opinion there. Are there other possibilities? Now, I had to put the names down there because I thought there's no way in the world I'm going to remember the names of the different berries that are available in Canada. And so that's the 4 on that side, and I'm sure you can pick those out much better than I can, but Saskatoon berry, wild lingonberry, buffalo berry, and choke cherry. And we arrived on Wednesday, and I looked at the local newspaper, and the headline was that choke cherry trees are being chopped down in Winnipeg because I've got some horrible sounding thing called a black knot. I've no idea what that is, but it really sounds terrible. But yeah, those are all ones that are at least semi-commercially available. I had a lovely fish on Wednesday night, trying to think which day it is. Arctic char with the Saskatoon berries, wonderful. But is it possible to take those and turn them into nutraceuticals, health foods, functional foods, whatever you want to call it? That's something for you people, obviously, to answer, not for me. But on the Australian side, Tasmanian pepperberry, the Davidson plum, and also the Illawarra plum are certainly local. They're Indigenous foods, but also Indigenous to Australia. So, yeah, all of the ones I've talked about in my Australian accent, all the ones I've talked about have been non-Australian foods. We're very good at looking at foods from overseas, and we have so little knowledge, in Australia at least, we have so little knowledge about the plants that grow in our own country, and I think we need that. Last slide. I haven't been stopped by the chair yet. That's also why I've kept him on that side, so I can't see what he's doing there. Okay. If we look at that, can we actually put together a diet that's not simply saying to a patient, go and eat 3 carrots a day, 1 for breakfast, 1 for lunch, and 1 for dinner? Can we actually make it a little bit more interesting? And yes, I think we can. And of course, these are all stolen from the internet. I'll get it right eventually. The purple plum juice is now available, at least in Australia, as a product. It's only been on the market about a month or so. But so that's a possibility as a juice. This is a purple carrot soup with some cardamom in it. I've actually tried that at home because making soup is within the limits of my cooking, and it was delightful. Even I've got 2 sons and 2 daughters. Even my daughters liked it. The salad with some of the different colours, but also some roasted beetroot in there. Very nice. The roti type food, and that one is one made with red sorghum. Now, sorghum contains the deoxyanthocyanins, among other things. We don't use sorghum as a food as a human food in Australia. It's for pigs and cows, but it is clearly the staple cereal in a lot of North Africa, also in North India. A lovely pie there that's with mulberries, also with anthocyanins. And then last but not least, close the meal off with a very good quality coffee. So it's probably healthy, but probably also tastes good, and that's an important issue. Thank you very much for your attention.
Good morning, everyone. So we are going to learn about purple phenolics from cereal grains and a little bit about their potential in health promotion. I'm a food scientist, so I am more concerned with the composition of the grains that we have in our food system, and also those that have potential that are not yet in the marketplace. So when you see the topic purple phenolics, what you think of are berries, especially here in Manitoba. These are Saskatoon berries, so they are a very good source of what we may call purple phenolics. However, in grains, you also find that some grains can be called pigmented grains because they have the color red or the color purple because of the presence of anthocyanins. We've had a chance to work with a few of the purple wheats that are pigmented, and from all over the world. We've worked with some. These are species from Denmark that we actually obtained from Denmark, but we've also worked with purple wheat that is coming out of Saskatchewan. With corn, there are some varieties which are very purple, like what Dr. Brown has shown, and this is one type of corn which we actually were looking at part of this summer, beginning of this year. This is blue corn, which is being grown in Italy. and that was at the dough stage before it actually fully matures and it's dried. So we do have among cereal grains potential to actually get some of these anthocyanins in the system. And I'm a big advocate of cereal grains because I look at grains as the main staples around the world. They are the major sources of our nutrients. And we now know that if we consume whole grain as opposed to refined grain, we can actually prevent some of the oxidative stress-related chronic diseases. So my focus has been looking at the dietary phytochemicals. We do have enough database on the general nutrients in grains, both the major nutrients and the micronutrients. So I've been focusing on dietary phytochemicals as bioactive compounds in human health. So when we are among nutritionists, sometimes we feel small because the grain, when you look at the small grain, it doesn't look as sophisticated as the human body. But actually, it is as sophisticated as the human body if you look at it closely in terms of the detailed structural organization. So this is an example of a wheat kernel. So you can see that there are different layers on the outside which are highly organized and highly differentiated. So if you peel from the outer layers as you go in, the outer layers, that's what we associate with the dietary fiber. That's where most of the fiber is coming from. And then the major portion of the grain is what we call the endosperm. So that is the major component here that is exposed, and it is at least 80% of the grain. And the endosperm is where we are getting the refined flour. We also have the germ, which is showing at the bottom here as the source of most of the vitamins and also the location of the lipids in grains. So we remove this portion because of the propensity towards rancidity. So to have a shelf life product that can stay for long, we remove the germ because of the lipids. So when we are talking about pigments in a grain, if we are looking at purple wheat, for example, you can see under this, with this cross-section that we've got a label which is called the pigment strand. So with the pigmentation that we find in cereals, most are located also in the outer layers of the grain, not in the endosperm. So that creates a challenge when we want to concentrate some of these pigments. So what we are getting from the cereals is mostly the nutrients, carbohydrates, which we divide into starch, non-starch polysaccharides, the protein, the lipids, the vitamins, and minerals. But we also are getting the dietary phytochemicals, especially the phenolics, and to some extent, especially with yellow corn, you get the carotenoids as well. So just the pains of trying to understand the botanical constituents of the grains in terms of the detailed chemical constituents. So we do try to section the grain so that we get the outer layers that we know in layman's terms as the bran and the endosperm, which is the major portion of the grain, and the embryo. So when you do that with rice, in this case we were using rice, you can see the different distribution, which is almost at least 80%, which is the endosperm. And then the bran and the embryo, they occupy a smaller component. So these are some cereals that we are looking at as containing these purple phenolics. So we have got red rice and black rice that are both being produced in China as the sources of anthocyanins. However, when we do measure the difference between the two you will find that it's black rice, which is a major source of the anthocyanins. So this is an elaborate classification that was done some time back of dietary phytochemicals. So when you are eating a diversified diet, you can actually access most of these phytochemicals. But when I'm looking at grains, the phytochemicals of major importance in grains are the phenolics and to some extent carotenoids. So phenolics, you can see that it's subdivided into several other subclasses. And in most grains, we can get the phenolic acids. So those are further divided into two classes, the hydrocinamates and the hydroxybenzoates. The other common phenolic are the tannins and the flavonoids. So when you are talking of the cereal sorghum, you can get a lot of these condensed tannins in some varieties. But for most grains, you are looking mostly at the phenolic acids and the flavonoids. And what we call purple phenolics are actually under the classification of flavonoids, the anthocyanidins among them. So phenolic phytochemicals can simply be classified as phenolic acids, and most of them, they are actually bound to cell structures in the grain, so they are not readily bioavailable. That distinguishes the grains from the fruits, because most of the fruit phenolics, they are soluble, at least 75%. The opposite is true for grains. Most are actually bound, and they are not readily available. And what we see as the pigmented grains, most of the color is actually coming from the flavonoids in the form of anthocyanins and the condensed tannins, the proanthocyanidins, which upon depolymerization can yield the anthocyanins. So there are a number of these compounds. If we are to understand the variation in color of fruits or color of grains, then we have to take the pains of actually determining which are the major anthocyanin in the plant that you are dealing with. So you can see that most will appear as esterified to a sugar. Esterified to a sugar, so those are the anthocyanins that we have in nature. The presence of the sugar will stabilize the anthocyanin structure. Whenever the sugar is removed, then the structures become more unstable, especially that in most of these foods, we have to do some form of processing. So these anthocyanins, they tend to accumulate during different stages of grain development. We were following with black rice, where we get the maximum accumulation of these anthocyanins. So you can appreciate from just the total anthocyanin content that after 2 weeks of flowering, 3 weeks of flowering, that's when we are getting the highest level of anthocyanins. But in a fully matured grain, we have reduced levels of these anthocyanins. We also look at what are the predominant anthocyanin compounds in each of the grains. So for rice, you have got cyanidin 3-glucoside and peonidin 3-glucoside. This will make up almost 80% of the anthocyanins that you find in rice. So we look at pigmented grains also as natural colorants, and on the market you may come across some which are some products like colored tortillas and snacks coming from blue and purple corn and red rice, which is also being used in bread and in ice cream and some liquor. So we want to look at anthocyanins as the main compound responsible for plant pigmentation, but also as a natural colorant and also as possible natural antioxidants and as anti-inflammatory agents. So when we look at a variety of grains which are actually colored with pigmentation, and you measure the anthocyanin content among those grains, there is a wide range of variation. You can see sometimes you don't even detect, even if the grain is looking blue, as in blue barley, but just looking across, you can appreciate not just the genotype variation, but among the cereals which are pigmented, there's a wide variation in anthocyanin content. So if we are going to reach maybe levels that we can use as possible for reduction of inflammation, for example, in this case, you can see black rice among these cereals that were compared as being the highest in content of the compounds. So when we process grain, the primary processing, we are trying to produce a flour. And by doing that, we can easily separate the embryo, the bran, and endosperm. And to get whole grain, normally these are combined back to produce a whole meal. But where you have got refined flour, for example, then you can look at the levels of the compounds that are remaining in each of those fractions. So when we do that, you can see that in the endosperm, we cannot even detect any anthocyanins, which means if you were having polished rice from black rice, then you are not getting the benefit of the purple phenolics at all. The embryo also is it's much reduced compared to what is remaining in the bran. So the bran has much higher levels of these anthocyanins, and the removal of the bran in terms of polishing, then we'll end up with no detectable levels at all. So we have also looked at processing, what it does in terms of these phenolics. So we compare brown rice. This is partially processed rice and red rice compared to purple wheat. So in this project, the graduate student was comparing those different pigmented cereals, producing infant cereals, and comparing the infant cereals with those that are already on the market. So I just want to highlight, not to expand on this one. The paper was published in 2011. That among the comparison she did, the purple wheat, which is coming from Saskatchewan, had very high levels of these anthocyanins. Even after processing to produce the infant cereals, you could still find significant levels of these anthocyanins. So this is homemade purple wheat infant cereal versus the lab-made purple wheat infant cereal. And on the other side, these are all commercial cereals on the market. What we found actually among those that we sampled among the infant cereals was that if we have got an infant cereal which has got some fruits, especially some berries. For example, this one labeled as H3 has got some raspberry. So you can still measure some anthocyanins in those cereals, but otherwise there aren't that many infant cereals where you can find measurable levels of anthocyanins. So in that study, we did conclude that the purple wheat, which is being produced in Saskatchewan, actually has very high levels of anthocyanins. Brown rice, we couldn't detect any anthocyanins, and then what we used was Heinz Stage 3 cereal, which was also containing high levels of anthocyanins. So we looked at the purple wheat, if it's transformed into beer, and measured also the antioxidant activity levels using the free radical scavenging assays. The beer is to be marketed as antho beer, showing that it still contains some anthocyanins. So we're looking at both the radical scavenging activity using DPPH of the beer that was produced, and with that, the production process, one used some hops and the other did not use hops. And by doing that, apparently just the presence of hops somehow reduced the free radical scavenging activity of the beer, the antho beer that was produced without the hops. So there are also quality differences that were detected in taste and aroma. We could distinguish the antho beers that were produced from the control beer. So we know that the brewing process also will affect both the quality properties of the beer and also the antioxidant activity in terms of the residual anthocyanin levels. So I'm going to talk a bit about these purple wheats that we obtained from Denmark. The last one, it's not a purple wheat. It has got more or less, it's more like common wheat. It was used as a control. So we had used the purple wheat from Saskatchewan before in a bread product, and in that project we could see clearly that purple wheat bread is much higher in its free radical scavenging activity compared to the refined white flour bread and also compared to the wholemeal bread. So we use a standard bread making procedure, which is the Canadian short process method that the Canadian Grain Commission uses. And we measure the anthocyanins that we extract in the flour during production and the end bread product. So when we are looking now at the total anthocyanin content, it varies throughout the processing. With the fermentation, the levels peaking during fermentation. Remember the other variety, which was a common wheat, it has no anthocyanin, and that is why the blue is consistently lower. So we do use different approaches for determining total anthocyanin content, and we prefer to use what we call the pH differential method, which is more accurate and also comparable to the total anthocyanin that is measured with the HPLC. So the antioxidant activity also follows the anthocyanin content that we measured, so they are also linked in some way. So I want to finish with the last few slides, looking at what has been reported in the literature on the effect of pigmented rice diet in animals with chronic disease symptoms. So this is just a summary of what I could find. So in this case, the conclusion was that pigmented rice diet generally lowered the chronic disease factor levels, and most of the rice that were being used were black rice, not red rice. In fact, some of the red rice, they do not contain measurable levels of anthocyanins. And then we have also been working with Dr. Gary Shen's lab in the Department of Physiology. We have been looking at the Saskatoon berry powder that we have prepared. We haven't gotten to the cereals yet, but the results are promising in the sense that you can see the effects of just the anthocyanins in the Saskatoon berry powder. And I'm glad that Dr. Lindsey Brown also showed some results on purple maize. And I want to think that we can repeat these same experiments using anthocyanins from purple wheat or from purple maize and be successful. So there is also another paper in press using the Saskatoon berry powder that is going to be released soon. So in my concluding remarks, I want to say purple phenolics are mainly associated with the anthocyanins, and the pigmented grains are a potential source of anthocyanins in the diet, and that the anthocyanin extracts of purple wheat, they are also always showing higher antioxidant activity compared to common wheat. And that the type of grain, the maturity stage, and the processing will all affect the levels of these purple phenolics. And that with time, I hope we will see a move towards getting dietary reference intakes for anthocyanins and other grain bioactives. So I just want to acknowledge some of the graduate students and the postdoctoral fellows who have worked on this project containing pigmented grains, and the financial support that's coming from the federal government through NSEC and the CFI. And Dr. Jones for inviting me to participate in this session. Thank you.
Well, thank you very much for the introduction and for the conference presenters to allowing me to share with you some of the work the Grain Research Lab is doing on barley, specifically with vitamin E. One benefit of being the third presenter is that some of the information that I was going to say has been covered, so I should be able to not be able to cover some slides a little bit quicker and get you to have lunch. I don't want to stand in between that. So the work that I'm going to be presenting today is a collaborative effort with Dr. Anna Badia, a barley breeder with the Canadian with Agriculture and Agri-Food Canada out of Brandon, Manitoba. And what she is wanting to do is develop food barley lines with increased vitamin E content. Green button. Okay. So just as a brief, I'm going to start by just giving a brief introduction about vitamin E, which will help set the stage when I talk about the content and composition that we find in barley and why this makes barley a unique source of vitamin E and potentially valuable as a potential source of vitamin E. I will then go on and look at the distribution where the vitamin E is found within the kernel, which is important when we are looking at the value-added potential of barley as a source of vitamin E with respect to pearling fractions, fiber fractions, and brewer's spent grain. So to begin, as we know, vitamin E is a lipid-soluble vitamin that is found naturally in the plant tissues. The natural form of vitamin E is made up of 8 chemical forms, and which I will refer to collectively as tocols. Within this family, there are 2 subgroups. We have the tocopherols, which will be abbreviated as T, and the tocotrienols, abbreviated as T3. And you can see within those 2 groups, the chemical forms are alpha, beta, gamma, and delta. If we just briefly look at the structure of vitamin E, tocopherol first, it has a chromanol ring with a phytyl side chain. It's very similar for tocotrienol, except the chromanol ring is, there's a unsaturated side chain where there's 3 double bonds. Where it comes into play with the different chemical forms, how they differ among each other is the number and the position of methyl groups substituted on the chrominol ring. For example, alpha tocopherol would have 3 methyl groups substituted onto the chrominol ring. If we look at beta tocopherol, we only have the 2 methyl groups substituted on the chrominol ring. At this point, I just wanted to take the time to just briefly explain the difference between synthetic vitamin E and these natural forms of vitamin E that are found as a natural source. And to do that, I just want to point out that tocopherol has 3 chiral centers. And without going into too much detail in the stereochemistry, what this means is that each form of the tocopherols, each form has the potential of having 8 optical isomers, which you will see as I do a comparison between synthetic and natural vitamin E. Synthetic vitamin E contains only alpha tocopherol, but it's a mixture of those 8 optical isomers that I spoke of, and they're listed here. Only these 4 are which are considered to be active forms for human. If we compare this to natural sources, plant sources of vitamin E, they contain all tocopherols, depending on the source. You can have various concentrations of the different sources, and they also can contain all of the tocotrienols. Now, in the literature, the first form of vitamin E to be recognized is alpha tocopherol, and therefore most of the research concentrated on alpha tocopherol. But and it's been extensively shown, the value of human health as an antioxidant is well documented. But as the research progressed, the attention has shifted to the other forms of tocopherol, including the tocotrienols. And these different chemical forms, it is important to point out that their bioactive activity is not redundant to that of alpha tocopherol, and in fact they are very functionally unique. Those distinct structural differences between the alpha, beta, and gamma and delta forms are very important in how, and have a significant impact on how they're metabolized by our body. So then we started to see in the literature titles like tocotrienols, health and disease, the other half of the vitamin E family, tocotrienols, the vitamin E of the 21st century, vitamin E, a dark horse, and the gamma tocopherol, the major form of the vitamin E in the US diet, deserves more attention. If I just bring you back to the structure, I just want to point out again that the main difference between the tocotrienols and the tocopherols was this side chain. And what this means is that there's a unique feature of this unsaturated phytyl side chain that provides easier access to the cells, better antioxidant functions, and activation of a wide variety of gene signaling, including cholesterol-lowering reduction. So there has been an emerging focus more on the tocotrienols as opposed to just that of alpha tocopherol. And here is just a brief summary of some of the health benefits, in addition, in comparison to just alpha tocopherol that we see. And if we look at the alpha tocotrienol, some of them are the cholesterol lowering and cardiovascular health, suppressing the activity of enzymes responsible for cholesterol synthesis, cancer prevention, prevention of neurodegeneration. So tocotrienols are being researched for benefits in bone health, blood sugar metabolism, and brain health. And the reason why I want to bring that to your attention is because there's various sources of these vitamin E chemical forms. Common ones that we're used to seeing is wheat germ, as well as other soybean, canola, sunflower, other vegetable oils, palm oils. But there's also cereals. These vegetable oils and wheat germs tend to be high in the tocopherol content and not so much so in the tocotrienol content. This table shows a comparison of the tocol concentration of some common whole grains. If I start first at looking at the wheat, we can see that wheat is very high in beta tocotrienol, as well as alpha tocotrienol, with a total content on average of about 60 micrograms per gram, keeping in mind that this will be very variety specific. But if we now look at oats, you can see that its highest is with alpha tocopherol and alpha tocotrienol, but the total content is a lot lower to that of wheat. Corn is unique in that it has a very high content of gamma tocopherol. And from that previous slide where I look at the health benefits in the table, you can see that gamma itself has its own nutritional benefits and importance in biological activity. But if we look at barley, barley is unique in that it contains all 8 vitamers of vitamin E, which makes it a unique source and deserves some attention. Specifically, the content that we're seeing with the tocotrienols. As the focus has emerged towards more looking at the health benefits of tocotrienols, this ratio of T3 to T has been a common criterion that's been used to assess the nutritional quality of a vitamin E source. This is some data that we collected from the varieties that we've been testing with Anabadia that looks at the tocol concentration for 26 barley varieties. Right here I report the range that we observed, and you can see in the total content, we see a fairly big range in the total content of vitamin E from 46 to 85 in the varieties that were tested. At this point, I would also like to point out that these 26 varieties were all grown in the same location, so any variability that we're seeing is a result of genetic diversity. And if we look more closely at the individual chemical forms, we can see a large range in that alpha tocotrienol from 22 to 48, beta tocotrienol, as well as the gamma tocotrienol. And if we then look more closely at the varieties that were tested, these included 16 food barley varieties as well as 10 malting barley varieties. And we did find that the food barley varieties were on the higher end. And I will point out that the one that had the 85 was actually a barley that was colored purple, had the purple color. This table looks more closely at 2 food barley varieties that have been bred and are commonly known to have high beta-glucan content. So they already have that nutritional benefit of having that higher content than other barley varieties of soluble fiber. But if we look more closely to what we saw when we tested these barley varieties at 3 different locations over a 2-year period, we can see that the content of the 2 barley varieties differed. We also always saw that the genetic variability of CDC Hylos to have the higher content compared to that of rattan held at all the locations that we were testing. Then the other distinct features between these 2 varieties is that alpha tocotrienol was higher in CDC rattan than CDC Hylos, and the beta. One feature that we were excited to see that was holding in all locations when we tested is that this CDC Hylos was very unique in that it had a very high content of the beta tocotrienol, the gamma tocotrienol. So what does this all mean? Well, it so far shows that there is the genetic potential to breed and increase the content of vitamin E in barley to further enhance the nutritional benefit of barley that we already know of based on its dietary fiber content. But I would also like to point out that there's only 2 to 3% oil in barley. So extracting oil from barley isn't likely a feasible task, but to our advantage, where the vitamin E is located is not uniform throughout the kernel, and it's found in the outer layers, in the germ and in the alurone and pericarp outer layers of the kernel. And this will bring me into a study that we did when we looked at the vitamin E content as we pearled off various fractions at a 5% level. So this is a CDC rattan. So this on the x-axis you have the content of vitamin E in micrograms per gram, and I've also showed the ratio of T3 to T for each of the fractions. We know from wheat germ that wheat germ is very high in tocopherol content, so we can also see that in barley. The purple portion of the bar represents the tocopherol content, and the green the tocotrienol content. As we move forward and pearl off the first 5%, we can see there's a shift. There's an increase in the vitamin E content, but also in the ratio of T3 to T. Our highest content of this variety was found in the next fraction, where we had almost 200 and I think it was about 90 micrograms per gram, with a very high content of T3 to T. As we move closer, we still get a higher ratio, but we start to get a decrease in the amount of vitamin E that we found. And then as we move further into the kernel and get closer into the endosperm, then we can see we're getting close to the results that we saw for the whole grain. So what does this mean? Well, you've probably all purchased pearled barley and pot barley. Well, this is a byproduct of that process, and if we can take advantage of the fact that these pearling byproducts are concentrated into that oil and either use that as a source of barley extraction, or given the proper food product, perhaps these fractions can be added and we can gain the benefit of the vitamin E content for these pearling byproducts. I would just, again, you saw this figure with Trust's presentation, and I would just like to draw your attention to the statement that I made, that the vitamin E is not, the distribution isn't uniform throughout the kernel, that we find it in the outer layers, the alurone, the pericarp, and in the germ. But that's also the thing that we find with other bioactive ingredients, that it's not uniform. So as a result, we can use dry fractionation technologies such as milling to produce fractions that are concentrated in the portions of the grain that are enriched in these bioactive ingredients. So at the Grain Research Lab, we have done extensive research in developing a mill flow in order to come up with a fiber-rich product, fiber-rich fraction that is enriched in these bioactive ingredients. This is a picture of a light micrograph of a barley fiber-rich fraction, which shows those components of the kernel that are enriched in the vitamin E, the fiber. The pericarp, as you see here, the pericarp and the alurone in the outer layers are very rich in a fiber known as arabinoxylans. Here is a picture of the endosperm. That's the center of the fragments of the center of the kernel, where you see in blue is the cell walls, and the cell walls are very rich in that beta-glucan, that component that barley and oats are known for. And they encapsulate the starch granules. So what happens during the milling process is that we're freeing up those starch granules so that we're removing the starch essentially, and we're leaving behind the pericarp, the alurone, that cell wall material that is rich in the fibers, and now that we know also in vitamin E. This table just shows a comparison of a common food barley variety, CDC Hylos, that looks at the whole grain and the fiber fraction and compares the different constituents. What I would like to highlight is that we see the decrease in the starch content, but we also see that increase in the beta-glucan and the arabinoxylan content, and also an increase in the vitamin E. We've done even further enrichments of these milling fractions and given the right variety, like if we do CDC Fabar, which as a whole grain tends to have a beta-glucan content of 9 to 10%, we can get fiber fractions that can be greater than 20% beta-glucan. And that would only also increase the content of, and the vitamin E content would likely also increase as well. The benefit of these fiber-rich fractions is that we can incorporate them into bread products. Either we've done it with pitas, with bread, with pasta, and that we don't have a negative impact on the quality of the product, because with these bioactive concentrations to be higher in content, we can add less of this fiber-rich fraction compared to if we had to add whole grain. And this is important, and as we know now, in Canada and the United States, foods that contain barley or barley milling fractions that contain 1 gram of beta-glucan per serving can have a health claim that they help reduce cholesterol due to the extensive research that has been done on beta-glucan. So having these fiber-rich fractions, we can add and meet those claims when you use a fiber-rich fraction over a whole grain. And just this is just a brief figure that shows a comparison of the vitamin E content to comparable products of the barley fiber-rich fraction, let's say wheat bran and oat bran. And you can see that Here again, the barley fiber-rich fraction has similar content to that of wheat bran, but you have all 8 vitamers present, which is unique to barley compared to other commodities and products. And then finally, I'm just going to briefly talk about brewer's spent grain, because it is a very, it is highly generous, there's a lot of volume of it due to the brewing industry. There's so much that is being produced of it, and essentially what's happening during that process is that we're concentrating that fiber fraction as well, similar to what's happening in milling, where we're removing that starch through mechanical processes. What's happening during the malting and brewing process is that enzymes are breaking down the starch and leaving behind the fiber. So this is a very nutrient-rich product that has great potential to be either used as a source of vitamin E extraction, oil extraction, or as a food product itself. This is just a figure that shows the comparison between barley and malt, and you can see during the process of making malt from barley, you don't lose any of the vitamin E, but then you see a significant increase in the content of vitamin E in the spent grain, and the ratio also increases with respect to the ratio of tocotrienols to tocopherols. And this is essentially because we've concentrated those parts of the grain that contain the vitamin E. So just in summary, the natural sources of vitamin E provide all chemical forms, which is different from that of synthetic. So it's important to find a good, valuable source, and barley is unique in that it contains all 8 vitamers in a favorable ratio. We do see that there's significant genetic diversity in the tocol content and in the composition, and that there is the potential to do through traditional breeding practices to further improve the quality of, nutritional quality of barley. And then finally, to realize the full potential, nutritional potential of barley, it's important to not just look at the whole grain, but also the byproducts that the industry can produce and what we can produce through milling and dry fractionation technologies. So thank you for your time, and enjoy lunch.